I'm going to start out the genetic technology chapter today by taking a look at section 14.3, which is the end of another chapter we've already studied, but we felt like this worked out a little bit better, made a little bit more sense with chapter 15 because it's a little bit more about genetic technology. So we're talking about human DNA. There's a ton of it. The smallest human chromosome contains 50 million base pairs. And in the human genome, there are actually 3 billion base pairs total. So that's every letter that there is, A matching up with T, C matching up with G, 3 billion of those in the human genome. In the 1970s, scientists found that when they're looking at DNA, they could break it up into pieces using enzymes found in nature. Using that, they could read the base sequences by using enzymes to cut, separate, and replicate DNA base by base. Nucleic acids are different from other macromolecules, and those differences make it easy to extract, remove from cells and tissues. DNA molecules from most organisms are too large to be analyzed, so they have to be cut. Bacteria produce enzymes called restriction enzymes that cut DNA molecules into precise pieces called restriction fragments. They're several hundred bases in length. There are hundreds of different restriction enzymes, and each one is unique. It cuts DNA at a different sequence of nucleotides. The EcoR1 restriction enzyme cuts at the sequence GAATTC. It cuts each strand between the G and A bases right here, leaving single-stranded overhangs, which we call sticky ends, with the sequence AATT. The sticky ends can stick to a DNA fragment with the complementary base sequence. So if you cut two different DNA sequences with EcoR1, they can join together because those sticky ends will base pair with one another. Once the DNA has been cut into pieces by restriction enzymes, scientists can use a technique called gel electrophoresis to separate and analyze the different fragments. This is what we're going to be doing in lab in the coming days. DNA fragments are placed at one end of a gel that's about the same consistency as Jell-O. When an electrical charge is applied to the gel, the DNA molecules which are negatively charged will move towards the positive end of the gel. The smaller the DNA fragment, the faster and farther it moves. So what you see on this graphic down below, the gels are loaded the DNA samples are loaded into the top of the gel. An electrical current is applied, and then the longer fragments stay more to where they started, and the shorter fragments move faster and farther. What we get out of this is a banding pattern based on fragment size. We can call it a DNA fingerprint, we can call it a banding pattern. Specific stains that bind to DNA make these bands visible. After these DNA fragments have been separated, researchers can read it or sequence it. Single-stranded DNA is put in a test tube containing DNA polymerase, the enzyme that copies DNA, along with the 4 nucleotide bases. The DNA polymerase uses the unknown strand as a template to make one new strand after another. Researchers add up some bases that have a dye attached. Each time a dye-labeled base is added to a DNA strand, the synthesis of that strand stops. When synthesis is completed, we get a series of color-coded DNA fragments of different lengths. So that's what you see here. These fragments are separated by electrophoresis. The order of the bands on the gel tells the sequence of bases in the DNA. This process is automated by computers. This DNA sequencing process can read thousands of bases in a matter of seconds. In 1990, the United States, along with other countries, started what we call the Human Genome Project. The goal of the project was to sequence 3 billion base pairs of human DNA and identify all human genes. Other goals included sequencing the genomes of model organisms to interpret human DNA, develop technology to support the research, explore gene functions, study human variation, and train future scientists. So the sequencing portion of this is done pretty quickly in this way. Researchers break up the genome into pieces using restriction enzymes. Base sequences in widely separated regions of DNA are determined. These regions are used as markers. The markers make it possible to locate, they're almost like bookmarks. They locate and return to specific locations in the DNA. Once researchers have marked the DNA strands, they can use what we call shotgun sequencing, rapid sequencing method that involves cutting DNA into random fragments, determining the sequence in each fragment, and then computer programs take the sequencing data, find areas of overlap, and then piece everything together by linking the overlapping areas. Computers align these fragments relative to the known markers on each chromosome. So what you see here on the right-hand side is what I'm describing. So DNA is broken into pieces, the computer sorts and orders it, the overlapping sequences are matched and aligned to determine the DNA sequence. Research today is focused on kind of interpreting the data from the Human Genome Project to look for genes and the sequences that control them. This is done by locating sequences known to be promoters. Remember, promoters are binding sites for RNA polymerase. This is how scientists can identify many genes. You can identify the gene, but then you have to figure out what protein it makes. Shortly after a promoter, there's an area called an open reading frame, which is a sequence of DNA bases that will produce mRNA. Other sites that help to identify genes are sequences that separate introns from exons, and then the stop codons, located at the ends of the open reading frames. You see all these here at the diagram on the right. We have the promoter, we have the intron, start codon, another intron, and a stop codon. So these give us clues that there's a gene in this area. If we were to compare the genomes of two humans, it's actually kind of crazy, but most but not all of their DNA matches base for base with each other. On average, one base in 1,200 will not match between two individuals. These single base differences are called SNPs, single nucleotide polymorphisms. Researchers have looked at these and discovered that sets of closely linked SNPs occur together time and time again. These collections of linked SNPs are called haplotypes, short for haploid genotypes. To locate and identify as many haplotypes as possible, the International HapMap project began in 2002. The aim is to give scientists a way to identify haplotypes associated with various diseases and conditions, pave the way to more effective life-saving medical care in the future. Copies of the human genome sequence, DNA sequence, and those of other organisms are available on the internet. Researchers and students can browse through databases of human DNA and study its sequence. More data is added to these databases every day. New field of study came out of the Human Genome Project called bioinformatics. It's the intersection of molecular biology and information science. And this is critical to studying and understanding the human genome. So this Venn diagram shows this area in the middle here, bioinformatics. An even more specialized field of study that came out of this is called genomics, the study of whole genomes, including genes and their functions. In June 2000, scientists announced that the human genome sequence was complete. First details appeared in February 2001. The full reference sequence was completed in April 2003, which was the end of the Human Genome Project, 2 years ahead of schedule. Human Genome Project confirmed that there are 3 billion nucleotide bases. This is interesting. Only about 2% of our genome encodes instructions for the synthesis of proteins, and many chromosomes contain large areas with very few genes. Seems kind of inefficient. As much as half our genome is made up of DNA from viruses and other genetic elements within human chromosomes. This chart compares human genome with other organisms. So here's our human DNA, 25,000 genes. Lab mouse. Arguably much less complex than humans. 900 fewer genes. Fruit fly, about half the genes. Just seems like humans are way more complex than to, if we look at these relative numbers, to be only 21,000-ish genes more to produce a human than a single cell bacterium. If we look at degree of relatedness, however, we see a lot of overlap. 40% of our proteins are similar to proteins in organisms such as fruit flies, worms, and yeast. Human Genome Project also pinpointed genes and associated sequences with diseases and disorders. It's identified 3 million locations where single-base DNA differences, SNPs, occur in humans, which may help us find DNA sequences associated with diabetes, cancer, and other health problems. The Human Genome Project transferred technologies to agriculture and medicine, and it started the U.S. biotechnology industry, fostered the development of new medical applications. So lots of different things have come out of the Human Genome Project in the last 13 or so years. Human Genome Project worked to identify and address ethical, legal, and social issues surrounding the availability of data and its technologies. Some of the questions are here. Who owns and controls genetic information? Is privacy different from medical privacy? Who should have access to personal genetic information? How will it be used? President George W. Bush in 2008 signed GINA, the Genetic Information Non-Discrimination Act, which prohibits U.S. insurance companies and employers from discrimination on the basis of genetic content of a human. The 1,000 Genomes Project, launched in 2008, is going to study the genomes of 1,000 people to produce a catalog of human variation. Data will be used to develop drugs, therapies, determine diseases, save human lives, hopefully, and preserve health. There are many more sequencing projects underway, an ever-growing database of information from microbial, animal, and plant genomes is expected. The most important challenge is to understand how all the parts of cells, genes, proteins, and other molecules all work together to create complex living things.